Thanks to Bioagnosis for the invitation and for hosting this to the Broad. I apologize in advance if I get a cough attack in the middle of this, but I'll do my best to be uninterrupted. I come from the other end of proteomics, which is you do all the discovery, you find all these interesting things. Spectronaut is doing a great job helping you guys with that. But what do you actually do with your findings? And SISCAPA is a technology that translates your findings into validated results and ultimately into clinical settings for going through regulatory submissions and beyond. So the first slide I have is just to make an announcement that as of last year, SISCAPA has joined forces with Bioagnosis as its exclusive CRO, realizing that we need a one-stop shop, a one-stop where you have the solutions all the way from discovery to targeted approach into validated assays. So we're combining the sensitivity and robustness and quantitative nature of SISCAPA, putting it together with the mass spec expertise and the software expertise that we have at Bioagnosis, and offering that as a one-stop solution to our pharma partners. For SISCAPA, I start with the name. It stands for Stable Isotope Standards and Captured by Antipeptide Antibodies. It's a mouthful, but it really drives home the message that there are only two components that go into a SISCAPA assay. There's the antibody, the SISCAPA antibody, and the internal standard peptide, which is used for quantification. Everything else in the workflow stays constant, and this is one of the major values of SISCAPA is once you implement it once, the only things that change are the reagents that you need for a given assay, and given that you're using a mass spectrometer as a detector, you can multiplex these assays any shape or form that you want. So we've applied this technology over the years to biomarkers, targets, and even therapeutics. There are papers out there where we've applied SISCAPA to measure therapeutic antibodies with specific idiotypes at very low levels in complex matrices. Like most proteomics applications that are bottom-up, SISCAPA starts with the digestion step. And that digestion step is actually quite important because if you don't do it, you have all sorts of non-specific protein interactions that give you false negative or false positive results. So by going through that digestion step, you're eliminating these kinds of non-specific interactions, but at the same time, you're making your matrix a lot more complex. You know, you go from 10,000 proteins to 100,000 peptides or more. And so that's where the role of the antibody comes in. We design these antibodies to be very specific to a proteotypic peptide from the target protein that we're trying to measure. And where the uniqueness is that these are very short linear epitopes. The shortest peptide that we've dealt with is a very good assay that we have for gelsolin, and the peptide is only 5 amino acids long. Taking you all the way back to your immunology class, an antibody epitope is usually 4 to 5 amino acids long. So we're specializing in making antibodies against very short epitopes that are specific to that and nothing else in the matrix that you're using for your measurements. And that antibody essentially goes into that very complex matrix, enriches the target, and you go through an extensive washing procedure, which means that your antibody needs to be high affinity, specifically low off rate, to be able to hold on to your target peptide while you're washing everything away. The end result is effectively a pure peptide sample of the target you're interested in measuring, plus the internal standard, which you use mass spectrometry to measure. So when I think of SISCAPA, I think about a hybrid between straight LC-MS and immunoassays. So like immunoassays, we are using an affinity reagent to capture our target, but that secondary antibody, which you normally have in ELISA, is replaced by mass spectrometry. So in doing so, we are harnessing the advantages of both systems. Like immunoassays, this is extremely sensitive, extremely high throughput, quantitative, and because we're using a mass spec for readout, we can do things like multiplexing, unequivocal identification, so on and so forth. So this is an actual SISCAPA run. There are different ways of implementing SISCAPA. There are those who take these antibodies, put it on a pre-analytical column in line with their LC system and do it that way. We prefer to do it on magnetic beads because ultimately these are for clinical implementations and magnetic beads are commonplace in the clinical setting. So what that antibody does essentially goes into that very complex matrix, enriches the peptide that you're interested in, giving you many thousands of fold of enrichment. And if you think about it, that means two things. First, you're increasing your signal. And second, you're decreasing your noise. And as a result of this, SISCAPA can deliver very sensitive, highly quantitative assays for clinical implementation. But that antibody does something, in my opinion, a little bit more important than that. It allows you to do what we call stoichiometric flattening. So because you control the amount of antibody, you can go in a complex matrix, enrich a little bit of very abundant biomarkers and all of your low abundance biomarkers, and essentially flatten that stoichiometry. So it no longer matters what is the dynamic range of your mass spectrometer. You can even implement this kind of a workflow on a MALDI-TOF with a dynamic range of 100-fold, as we have done in the past. So that stoichiometric flattening is a crucial advantage that you have, and therefore you can multiplex these assays in any shape or form. So we internally regularly measure albumin, which is the most abundant protein in plasma, next to super low abundance cancer markers, and we can do so in the same sample in the same run at very low cost. So I'm preaching to the choir here, but proteins are a lot more interesting than your genes. The way I think about it is that your genome is like a sheet of music. It's very sort of in place and very fixed in space and time, whereas proteins are like improvising as you're playing a sheet of music. And so you can have many different variations, and that is what, in my opinion, makes proteins very interesting. But there's some key questions that one needs to ask about proteins. And one of them that we are interested in, because we are a quantitative assay or technology, is how is the abundance of proteins in circulation in our bodies related to our genome? Is there any correlation? So a number of years ago, we got our hands on 20 pairs of monozygote twin samples and started asking this question. Is your genome determining how much of a given protein is in circulation in you? And the answer is, for some proteins, yes. So here's an example of mesothelin. You can see there's a pretty good correlation between what your genome dictates and how much of that protein you have, unless if you develop disease conditions like mesothelioma, and then that's easily detectable. But what is interesting is that for most of your proteins, that is not going to be the case. So yes, there are those that are strictly controlled by your genome, but pretty much everything else is not. So you could be sitting next to a person who has an identical genome as you do and yet have a very different protein profile. We call this the protein fingerprint. I don't get to go into that story today, but essentially each of you has a unique protein fingerprint different from the person sitting next to you, even if that person is your identical twin, and that's dictated by your environment, by your everyday life, what you do, what you eat, how much you exercise, so on and so forth. So of all the different sample types that one is interested in, we are primarily interested in plasma. We call it the deepest proteome. It has a dynamic range of 10 to the power of 11. Pretty much everything we ever wanted to see, we can find it in plasma, and I think you guys agree with that. SISCAPA is a sample agnostic technology. So we've so far implemented this in plasma, serum, whole blood, CSF, urine, dried blood spots, so on and so forth. So the reason plasma is interesting is that it essentially has a snapshot of everything that's happening in all of your tissues. It's like a river going down through a city and basically picks up bits and pieces of information from different parts of the city, So if you could really get deep into that plasma, you will find interesting things. Now, at the moment, what I'm hearing in this meeting as well is that you're comfortably able to measure about 10,000 proteins in a given sample if you use technologies like the P2 by diagnosis. So when you plot that, and this is a famous plot by Lee Anderson that keeps getting updated from time to time, but you see that that dynamic range of 10 to the power of 11, you can measure things with a mass spectrometer sort of right to the middle. And these mass spectrometers are getting fancier and more expensive, but they can give you better sensitivity. But the question is, what about all the markers down there that you're not able to see reliably on a mass spectrometer? I find this plot really interesting. So this is essentially tracking how many proteins were known to us as a function of time up until 2010. And you can see that we started in the 1930s with a few 1D gel type experiments. And in 2010, and by then mass spectrometry was a thing, and people were measuring proteins with mass spectrometry, but we could reliably measure about 1,000 proteins, which was roughly the same order of magnitude as we could see with 2D gels. But clearly, over the past 10 or 15 years, as we've heard today with the DIA technology and software that allows you to dig deeper, we can reliably measure more than 10,000 proteins in a given sample. That is really exciting. But it also causes a major problem. Why? Because if, the last time I did a PubMed search, there is more than 3,000 putative biomarkers for cancer in the field of oncology. And yet, if you actually count the number of proteins that have been approved by the FDA for diagnostic use, you can just count them on your 10 fingers. So there is clearly an issue here that proteomics as a whole, discovery, is doing a great job finding all these putative biomarkers, but there's no one there actually translating this into clinical use. So I would call that problem number one. We have an interest in diagnostic as the other side of our business. And we have identified a different problem, which is even when you're measuring these proteins clinically, the end result is a binary result. You're either normal or abnormal. But in fact, we are taking out one of the most important variables out of diagnostics, and that's the variable of time. So if you track a person as a function of time, where they are on that normal distribution can change. And those changes are absolutely meaningful when it comes time for you to diagnose disease or track treatment options, so on and so forth. So the whole idea behind SISCAPA, and this is now a little over 20 years that we're working on this, is that you do discovery with thousands of proteins identified, and then you need to have a high throughput, sensitive, quantitative system that can validate, take you through clinical trials, and ultimately for clinical implementation. And that's where SISCAPA comes in, and that's where we've been working with our pharma partners on. But we also have an internal R&D sort of work that we do. And this is something that a lot of people haven't heard before, so I thought maybe I introduced the idea to you. So with high throughput, you can go through thousands of samples and do your validation and your clinical trials and everything, right? But there's another side to high throughput. High throughput means that you can measure many samples from a single individual and bring the function of time back into diagnostics. So we started applying our technology to dry blood spots and started essentially looking at a number of interesting panels, from inflammation to iron metabolism to cardiovascular disease. And we started essentially doing these kinds of studies more than 15 years ago. So basically, people who work at SISCAPA, I consider them to be the most sampled human beings on the planet. These are people who essentially sample themselves every single day. They wake up, they have a cup of coffee, and they take a sample. And we call that your sample diary, hence the name of our device. because the idea is that you're taking a snapshot of your health for that day. And so we heard an interesting talk about measuring turnover rates in vivo. When you measure the turnover rates of proteins in vivo, you realize that some of them change on the order of minutes, 10 to 15 minutes. Others, like hemoglobin, take 3 months to change their concentration. So this is why we wanted to start measuring things on a daily basis to see what's happening in us. So this is the very first experiment we did in dry blood spots. And these are daily samples that we were collecting. And we were really excited. This is the inflammation panel. We could identify when the person had a nose cold versus pneumonia, and there was a meaningful change in the level of inflammation in that person. Well, that is not exactly revolutionary when you think about it. Pneumonia is a lot worse than a nose cold. But then we observed something really interesting. When we started zooming into the baseline, we were amazed to find that these inflammatory response proteins are going up and down together harmoniously. So the probability of that happening at random is infinitesimal. These proteins are actually responding to everyday life, depending on what you do, and they go up and down together. And then when you actually get ill, that's when you see those massive responses. So the other sort of interesting part, which I'm not going into, is that these little biological ups and downs is unique to the person. So yours ups and downs are going to look different than mine, which is absolutely fascinating. But then we started to do this on more interesting cases. So here's a case of a hip replacement surgery. What you can see is that before surgery, the person had a higher level of inflammation. And then post-surgery, sort of 3 months after, they have a new baseline, which is about half of what it was. So surgery was quite successful. But you can also see that the person had a nose cold, 2 months out, and the same proteins that react when you have hip replacement surgery also go up and down when you have a nose cold. These are inflammatory response proteins. They're non-discriminate. However, when you put in the function of time, the rate at which those proteins change is different, depending on the condition. And therefore, you can create personal health models. We call these the loop models that can differentiate between something like common cold versus surgery versus pneumonia. And the reason we call these loop models is the number on the graph is days. So basically, when the loop's starting to go back to the baseline, that's when the person is recovering. So in the case of a cold, it took them, about 4 days to start going back to the normal. Surgery, it took them only about 3 days to go back to the normal, so on and so forth. So when you put in the function of time and you increase sampling of a given person, you're able to start making these kinds of health models with seemingly non-discriminate proteins. We do a lot more of these studies. For today, I just picked a couple of interesting examples to share with you. But the other topic I wanted to discuss is one of sensitivity. So because you have that enrichment step, you're essentially delivering pure sample into your mass spectrometer. In many cases, we even don't use a liquid chromatography system. We just go direct injection into the mass spec. So when you do the math, basically, with SISCAPA, we're now, when you combine it, especially with the expertise at Biognosys, we're down to the femtogram per mil range for a lot of these assays that we're doing, which gives you even better sensitivity to most immunoassays. So the example that I have for you is the case of a protein called UBE3A. I'm not going to go into the biology of this, but this is the target for a monogenic disease called Angelman syndrome. And it's interesting because you get one copy from mom, one copy from dad, but in our neurons, for some reason, the dad's copy is silenced. So in all of us, UBE3A in our neurons is only produced from the maternal copy. Now in these patients, that copy is mutated or you have some sort of deletion, and therefore the protein is not present in these patients, which leads into a lot of developmental issues. So what we have done in this case, there were a lot of sensitivity issues with this assay. We developed a SISCAPA antibody for UB3A coupled to magnetic beads. Now, where diagnosis comes into play is a fully automated system which they have running the SISCAPA workflow. And remember that the workflow doesn't change. You just change what antibody and peptide pairs you put in there. And they've coupled that to an EvoSep connected to an Explorers 480. So this is as sensitive as you can get with mass spectrometry. And what they were able to show is that the LLOQ of this assay is 160 femtogram per mil. That translates to zeptomoles on column. And just a reminder, a zeptomole is a place between attomoles and anomoles. That's it. That's the end of sensitivity. You're essentially counting molecules at that point. So what we were able to show is that in neurotypical patients, CSF samples that were collected post-mortem from age 54 to 84, you get a very nice peak for UB3A. It's very measurable. And that is to be expected because all of us have UB3A in our neurons, and some of that leaks into the CSF. Now, what you would expect is that in an Angelman syndrome patient, you wouldn't see that. Why? Because the maternal copy is mutated or has deletions, and therefore you don't expect to see a lot of the protein. And so we, in a collaboration with the Foundation for Angelman Syndrome, we got some Angelman syndrome samples, and that's exactly what we saw, which is in these patients, we're not detecting a lot of UB3A, and because this assay is so sensitive, you know, we are sort of estimating below LLOD, but we are still able to see little peaks here and there. But because we are able to multiplex the SISCAPA assays, we're able to, for example, look for hemoglobin to see if there was any red blood cell contamination. We are able to look at myeloperoxidase at the same time to see if there's any neutrophil contamination in these samples, you know, which could lead to a false positive result. So the combination of the two platforms, mass spectrometry expertise, software from Biognosys, and the SISCAPA workflow, we are now comfortably able to deliver femtogram per mil, sort of low attomole, you know, mid-level zeptomole level sensitivity for these things. So SISCAPA obviously develops custom assays. So if, you know, the result of your discovery is some unique protein for which we don't have an assay, we leverage our pipeline to create a de novo assay for you. But we also have a growing menu, which currently has 150, 200 different assays, a lot of them clinical assays, which people use for patient stratification, sort of clinical trial type stuff. But we have a growing list of research assays as well, especially in the neurodegenerative disease space, in cardiovascular and oncology. Those are three areas that we are starting to develop more and more pharma-based assays. So if an assay is already developed, the whole thing is completely de-risked for you because we have the assay, Biognosys has the workflow, and essentially you just send your samples and you get them analyzed. So on that note, I want to just give you an overview of SISCAPA assay technologies. There are three lines of business that we are active in. Our pharma and CRO work through Biognosys, and you know, we basically work with big size, medium size, small size pharma, especially in small size on gene therapy and sort of small molecule drugs, and with pharma, more traditional types of drugs. And we have done so basically since our inception. But we also have clinical SISCAPA assays. So SISCAPA is currently run clinically in 16 different clinical labs from Europe all the way to Asia, with large reference labs in the States like Mayo Clinic, Quest, ARUP running SISCAPA for a test called thyroglobulin. And more recently, since the pandemic, we've deployed SISCAPA as a rapid deployment tool in support of pandemic preparedness. So we developed gold standard SISCAPA assays for COVID that were used in the UK, in NHS, As part of their screening program, we developed assays for RSV, AB, influenza AB, that they're running in Europe for pandemic preparedness. More recently, we have developed some gold standard assays for detecting neonatal forms of syphilis. And this is a growing area, something that we are quite interested in providing a service. So on that note, here's a team at SISCAPA. Most of you know Lee. He's been in proteomics for a very long time. He's published a lot. And I would also like to mention Terry Pearson by name. He was my mentor and PhD supervisor 22 years ago, and we were working on the very first cases of how do you actually make an antibody against a very short peptide that has a linear epitope. So I was working with Terry on that problem. So on that note, once again, thanks to Biognosys for the invitation and to Broad for hosting this, and I'm ready for your questions.
Great talk. I have a couple of questions because I've been using this a few years ago, similar at Pfizer. When you said that your antibody will be actually using magnetic beads to pull down, so they are biotinylated, or how you pull it down?
No, we find that biotinylation actually reduces the performance of the antibody. So these are cross-linked to protein G, which are immobilized on the beads.
Perfect. And if you have a new epitope, how long it takes for you to do a de novo?
It depends on the model system that you use and whether you want polyclonals or monoclonal-based reagents. We recommend rabbits because as legomorphs, they have delayed affinity maturation. On average, they give you better reagents. So for a poly, it takes about 2 to 3 months, and for a monoclonal, about 5 to 6 months.
Thank you.
Yeah.
How would you contrast your technology compared to Olink, who is also using the antibody-based?
Yeah, absolutely. I'm thinking whether I give you the unhinged answer or the political one. This is a quantitative technology used in clinical settings. With Olink, you don't have absolute quantitation. So that's the major difference, number one. And the evidence there is that when you compare Olink, SomaLogic, P2, mass spectrometry, the variance that you see in the results is staggering. In the early days of Olink, we took our menu, our entire menu, and asked the question, which one of these can we measure with an Olink panel? And the answer was none of them. And we think that might be deliberate because if Olink actually put proteins on there for which clinical grades assay exist, then they would get the kind of comparison that they don't want to see on the quantitative front. So that's difference number one. Difference number two is this is a peptide-based technology, and that digestion step up front gives you quite a bit of flexibility because you can use different kinds of proteases, and you can have different volumes. So the technology goes from 10 microliters to 400 microliters in a 96-well format, gives you immense amount of throughput and flexibility on how sensitive you want to get on an assay. So we view Olink, I put Alamar, Quanterix in the same realm. They're semi-quantitative discovery, at best targeted discovery platforms. This is a super targeted clinical grade platform that gives you absolute quantitation that you need for clinical trials and if you really want to know how much protein you have in there. Yeah.
I was curious if you've done any looking into cryptic exons in the context of neurodegenerative disease and quantifying it?
We have not looked into cryptic exons, but I can tell you that we have talked to a lot of people who have done their discovery, and surprisingly, the list is very small for what a neuro panel is going to be. Everybody's interested in tau, brain-derived peripheral form, A beta 4042, GFAP, NFL, and a couple of additional targets. So we've decided we're just going to make the whole panel. And within the next few months, we're going to have a neuro panel. We currently have alpha-synuclein, LRRK2, and tau is about to be finished. But, you know, this is what I was talking to Jan earlier, is we want to know what you guys find from your discovery work. This is, I think, where that translation is not happening, is that discovery takes place, and you publish, and the discovery papers are usually, oh, we found 5,000 proteins, but nobody actually narrows it down to a list of, here is 100 that really gave us, we are really interested in these 100. And then from there, it's easy for SISCAPA to say, we're going to make research-grade reagents quite fast and inexpensively, look at all the 100 in a high-throughput fashion, and this narrows down your list to now 5 or 10 assays, and then we can go and develop a gold standard set of assays for those. So this is essentially the transfer that we are trying to streamline with Biognosys. There is now a solution that you can take your list of 10, 20 to 100 assays and narrow it down to the top set of markers that you want to translate into clinical use.
Thank you, Morty.
Thank you.